Welcome, everyone. I'm Suzanne Kincaid. I'm the Chief Operating Officer of Aperio Clinical Outcomes. We're a customizable CRO based in Morrisville, North Carolina. My background, I've been in industry about 24 years now, mostly oncology. I was with AstraZeneca for a number of years in their early phase oncology division, developed a drug for Protherix in the oncology space, and then been with a mix of pharma and CROs for the last several years. So I'd like to introduce all of our panelists. I'll start with Dr. Brian Formansky.
Yeah, hi, I'm Brian Formansky. I'm Senior Director of Clinical Pharmacology at Nuventra. Before that, I was a Senior Clinical Pharmacology Reviewer at FDA, and prior to that, I was at GSK.
I'm Todd Shearer. I'm Senior Director of Clinical Pharmacology at SpringWorks Therapeutics, a small biotech here in the Durham area, also up in Connecticut. I was with Nuventra previously for 3 or 4 years, and prior to that with GlaxoSmithKline for about 7 or 8 years, and a few others before that. It's a long enough history.
Hi, I'm Patrick Roberts, Senior Director of Translational Medicine at G1 Therapeutics. Following the pharmacy school, came down to UNC for their translational research program, and then found my way to Ned Sharpless's lab for a postdoc. And when I met with Ned, he said he had this great idea of how we could protect patients from the side effects of chemotherapy, and that would be my postdoc project. And so that was about 11 years ago. And from that research, G1 Therapeutics kind of sprang out of UNC, and here we are today.
Thank you, gentlemen. So I want to talk about what we're going to be discussing today, but before we do that, I want to let you know that Brian and Nuventra are going to be having open office hours right after this session next door. So if you have questions and you'd like to ask Brian a few questions, especially with his FDA reviewer background, he's a great resource to go to. So looking at our agenda for today, we're really going to talk for just a minute about the landscape of the industry currently, and then we're going to do a few topics. We're going to talk about the regulatory strategy. We're going to talk about what to think about when you're planning your first in-human clinical trial, how to assemble your team, and then operationalizing your protocol. So a lot of topics to cover today. But just thinking about the landscape of where we are right now, it's getting harder than ever to develop drugs. If you look at the metrics right now, we know it's costing approximately $2.6 billion to develop a drug today, and over 10 years. And it's not getting better. In fact, it's actually getting worse. Ken Getz, with the Tufts Center for the Study of Drug Development, says we're no faster at developing drugs today than we were in the 1990s. Only 1 in 10 drugs ever makes it to approval. And our failure rates today are at the highest rate they have ever been. When we talk about oncology, because most of our panelists have oncology experience, only 5.1% of oncology drugs ever makes it to approval. But who is developing the drugs is, I think, even more important. It used to be big pharma, and as you can see, year over year, it is small companies. There was a great article in Forbes in January by Bernard Munoz that kind of broke down the 2018 FDA approvals. For the 2018 FDA approvals, 70% were with small companies, and 60% were with companies that had never filed before for an approval. So what this means for our audience and for you and for me is that in big pharma, we were surrounded by experts that could help us when we were ready for our first in-human trial. We were surrounded by toxicologists, epidemiologists, pharmacokineticists, statisticians. Now it's 2 or 3 people with a concept, very promising potential compound, and nowhere to turn. So we're here to talk about how we can help you eliminate the white space, all those days and weeks, the days and weeks that you're not putting your patients into a trial, and help you begin with the end in mind. So we'll start with assembling your team, and I want to ask each of our panelists, with that in mind, what would your bare minimum team members be in your very earliest planning phases? Brian, let's start with you.
Yeah, I would have to say probably a clinician, someone who has an MD background, and then a business guy, right? Because you need a lot of money to develop drugs.
I think it's good to take a soul search of what your own expertise is, and think about how other people might be able to complement that. Seek out people who aren't like you, who don't have the same expertise that you have, and pull those groups of people in. I think you've got to have tox covered, you've got to have CMC covered, you've got to have the clinical, and you've got to have the money. I'm sure I'm leaving one out, but pharmacology. But again, reach outside your comfort zone and bring in people that aren't like you.
Yeah, I think I would just echo the same sentiments. You know, our founder was a physician scientist, so we had the MD. I had clinical experience from working in the thoracic oncology program at UNC, so that helped, particularly when we decided to go into lung cancer for our first indication. And then we were really fortunate that at the time GSK, as we were getting started, laid off a bunch of really great scientists. So we got two great drug developers with 15 and 20 years that were willing to move into a tissue culture lab at UNC with no windows and work about this close together for 4 years. And they were great because they had done, you know, they had been part of teams and knew what it took. And then we were able to fill in the gaps. So we had great tox consultants and DMPK consultants in the early days. And then the next one, I think, once we started to think about going into patients, then CMC became the next kind of critical step.
Todd, what expertise do you hire versus use a consultant for?
I don't want to reiterate what I said, but I think you have, if it's an expertise, if it's something you're really uncomfortable about, you know, one way to figure that out is, you know, really study the guidance. You know, the guidance put out by the FDA will give you a step-by-step plan of what you need to do to get to an IND. If there's areas that you're really uncomfortable with, those are the experts that you need to seek out and pull into your team.
And for Todd or Patrick, where do you go to find the expertise you need that's very specialized, like DMPK?
Yeah, well, for us, being in this area, we were able to just kind of tap into our network. So there were, and once we got Jay and John from GSK, then they had other folks that had left GSK and either started their own companies or gone to specialized CROs. Of course, UNC has a great kind of tech development program of themselves, and so we were tapped into that, and we were looking for funding and NC Biotech, and so we started, you know, talking to folks and coming to events like that, like this, and networking, and that really helped us identify folks and fill the gaps as we were growing and our needs started to change.
Brian, what are the key disciplines you feel like you need for a first-in-human trial? And I think you had a really good story about engaging with a clinician.
Yeah, so the key disciplines are toxicology, CMC, and obviously clinical piece is huge. And so you have to understand where you're going, and that I think is where the clinical piece comes into play. But then you have to make the drug, right? And that's usually what holds most people up, to be honest. That's the most common reason people go on hold is because of CMC manufacturing issues. And then toxicology is actually the next one. If the toxicologist doesn't agree with your toxicology package, then you're going on hold too. And all those activities tend to take a tremendous amount of time to release those holds typically. So 6-9 months.
I would just reiterate the CMC piece. It's not an area that I'm an expert in, but it is probably the biggest, most underestimated chunk of work and money that anyone can. I think a lot of us come in with that pharmacology background, or you have the physicians, or you can run clinical operations, but being able to make the drug at scale, at scale above what you were making in your little 10 mgs or 20 mgs in the hood in the lab, when you have to make kilograms, tens of kilograms, hundreds of kilograms to support a clinical trial, it's quite a different matter.
Great. So Patrick, what's the first conversation that you need to have about regulatory strategy?
Yeah, so for us, we were developing a compound that protects patients from chemotherapy. So we're going into an oncology setting, but we, had some real critical clinical pharmacology questions that we were wanted to answer so that we could establish the right dose for going into patients. So the first question that we asked ourselves was, are we going to do a healthy volunteer study or are we going to do a patient study? And how is that going to look? And that really is critical because it gets into the toxicology and going to speak to the FDA because their main concern for that first study is safety. And so if you're talking about a single dose escalation, that's one tox package that you need. But if you're talking about continuous dosing or multiple doses over multiple cycles, then that may require a different tox package. So that was a critical piece and something that we kind of settled on ourselves, went and had a pre-IND meeting with the FDA to discuss, and then ultimately decided to do a healthy volunteer, and we did that outside of the U.S. and then brought that data back. But that was a critical discussion and decision for the company to make.
Brian, having been a regulatory reviewer, I'm sure you have some thoughts on what should your first conversation about regulatory strategy be?
Yeah, I think as the quote says, there tends to be a lot of focus on efficacy. Yeah, and I think it comes from investment perspective. You want to make sure your drug works and have confidence in moving forward translationally. But from a regulatory perspective, you show it once and that's pretty much good enough. Whereas the main focus, as Patrick was alluding to, it's safety. All they care about is safety for your first-in-human dose, and so it's a real big conversation between the toxicologist and the clinical pharmacologist, and who or what your first-in-human dose will be. A lot of it's dictated by the toxicology package you have, but then sometimes you'll need modeling and simulation to help justify the dose if you're trying to move that dose away from a MABEL or a Noel, which are more conservative dosing strategies than something like an HNSTD10, which is more commonly used in oncology.
Patrick, what are some of the pitfalls of your regulatory strategy? I know you guys decided to go to the Netherlands for your first in-human study. So some of the pitfalls or some of the unexpected things that can happen no matter where you run your trial.
Yeah, so like I said, we had this debate about do we go into humans first and try to dose escalate. We had theoretical concern that at low doses the drug may not work and the tox might be bad, so we thought we really needed to dose escalate in healthy volunteers, have a pharmacodynamic readout that really makes us feel assured that when we go into patients that we're at least starting at a dose that's not going to do any harm. So when we did that, that pharmacodynamic assay became really critical for that first-in-human healthy volunteer study. So there was a phase 1 site in the Netherlands that had the capabilities to do the bone marrow flow cytometry that we needed to do on site. And so the plan was that we would go in using the Noel and start low and dose escalate. We had some other pharmacodynamic markers during that study, and once we thought we were at the biologically effective dose, which was also supported by PK/PD modeling from Nuventra, that we would then expand at that dose and then do these bone marrow aspirates the site there had all the capabilities and really sold us on doing that. So we got there, we filed, we were all ready to go, and the health authority there said, well, everything looks great except for we're not sure that it was really justified to do bone marrow aspirates in healthy volunteers. And we were like, well, that's the whole reason why we're here. What do you mean? So they said, well, we want you to dose escalate, bring all of your other PD data from your other assays to us, and then write a justification for why you still need to do that expansion cohort, which was really the whole point of the whole study. So, it was a little bit of fingers crossed, do it, and hope that that we could write a good justification, and it ultimately worked out.
Yeah, I think one of the things that's interesting about the first-in-human dose to me is that's where all the disciplines come together. You have toxicology, which has to play into, is this going to be a safe dose? Do you have enough safety margin? You have pharmacokinetics and drug metabolism and pharmacology coming together. Is this a dose that we can achieve? Is it going to be an efficacious dose, especially if we're going into a patient population? And then you have to have CMC be able to support that. Do you have a solid tablet? Is your dose going to be 1,000 mgs, or is it going to be 2 mgs? Because that's a very different approach from a CMC standpoint. And so you've got to have some idea when you're starting those studies, how much drug substance do you need? You're going to start at 1 mgs, but you really need a 1,000 mg dose, ultimately you may not be able to support that if you've been planning for a lower dose all along. So I think that's where, to me, it's key to get a handle on what, not just what your first safe dose is, but what your first efficacious dose may be, because that's where you're going to spend the rest of your clinical development.
Right. And I think you don't want to have delay. So if like you said, if you start off making low dose tablets in 50s and 100s, but your drug looks great and your dose escalating to MTD, and all of a sudden you're at 800 or 1,000, and now you're trying to give people 10 tablets a day, that's a little bit of an issue. So then you got to go back and make new tablets, which comes with a whole other set of regulatory and CMC issues that potentially can delay enrollment as you're escalating, I guess.
For years,
Yeah.
not a short period of time.
Well, it's very similar with biologics too. The establishment of a MABEL dose is typically your first dose for a biologic, but a lot of people try to position the dose a little bit more aggressively, and then the FDA will come back and say, no, we don't agree with your dose, we want you to start at a much lower dose. And so to support that analytically becomes quite challenging, and people will go away for several years trying to figure out how to support that and deliver the dose they're expecting to deliver at such a low 0.001 mg per kg dose level.
Because you can't measure it in the dose
Because you
solution.
can't measure it, exactly.
Brian, for all of our audience members that don't know all the acronyms, could you just define for them what
sorry.
HNSTD, NOEL, and MABEL mean?
So MABEL is minimally active biological effect level. NOEL is no observed effect level. No adverse effect. Yep. And then HNSTD10 is highest tolerable.
Highest non-severe toxic.
Yeah, there you go.
Very good. Didn't know I was going to ask that.
Together we can get it.
There you go.
Between the four of us, we can. Brian, how do you prepare for your first in-human trial?
Yeah, it's a first in human study, as I was alluding to, it's a lot of disciplines coming together. And it depends on what your team is like, right? And you have to understand where your strengths and weaknesses lie. And you have to understand that you may not be able to clearly articulate your message to the FDA. So a lot of that, you end up having to hire medical writers and toxicologists. I mean, it's a big team effort for a first-in-human study. And I think preparation-wise, it takes a lot longer than people expect. Putting an IND meeting package together, it's 6-9 months, sometimes longer. It takes a considerable amount of time, money, and effort to bring all the pieces together. And any one of those pieces that's deficient can result in delay of your program.
Todd, what about you? Preparing for your first in-human trial.
I think it depends a lot on where you're starting from. We're speaking in generalities that we're starting from scratch with a new molecule that has never been in a human before. Sometimes you may have a molecule that's been in other patient populations, but you're looking to start a new IND. So you may not have as much to fill in that cases, but there may be also baggage that you need to clear up with that molecule or to risk the discharge. So I think it depends on where you're starting from. If you're starting from scratch, I think Brian covered it pretty well. It does take a while. There's a pretty straightforward path to get there. It's spelled out. You need to cover all the bases and hire a good team.
Patrick, what about you in preparing for your first in-human trial?
Yeah, I think the thing that I would add that I thought was really valuable is that we prepared a pre-IND package. We brought our tox consultant with us, who's been in front of the FDA, who's been on teams talking about hERG signals and whether hERG studies are, whether we should even be doing them at all. And so he had a lot of experience where most of our team had never been in front of the FDA. So it was great just to have him there as a presence, but also since establishing the dose and tox is one of the main concerns, then he was really the focal point. So even though he was an external consultant, he was critical, and he's remained a consultant for us over the last 10 years as we've everything we needed for the first program, and then we've brought him on or kept him on for all of our other programs. So And I think that's the only other piece. And the pre-IND meeting was great because then you get to start developing that relationship with the agency. You get feedback from them, and then you can, if you've given yourself enough time, you should take their feedback and then modify it as needed.
I just add one more thing. I think it's good that we haven't brought up the begin with end in mind, I think, yet. I think it's good if, as you're starting out, to think about what your clinical study is going to look like. Is it going to be in patients or healthy volunteers? How long is it going to be? Is it a short-term treatment? Is it a chronic treatment? Because that will play into how your entire CMC development goes. Your entire tox package may be different depending on what you're going into. So I think you really need to outline what that clinical study, even if it's early days, is going to look like before you can begin to prepare. You could be running 1 week or 2 week long tox studies, or you may be running 6-9 month long tox studies. And it's a big difference in the effort. Sorry.
Yep.
Anything you wanted to add, Brian?
I would just state that if you do go to the FDA, it's advantageous to have as much information as possible. Having a protocol synopsis can get agreement on your clinical study design, which makes it a lot easier for when your first-in-human study is submitted as an IND, as a new IND. It becomes much more of a checkbox exercise to make sure all the agreements that you had in your pre-IND meeting were incorporated into the IND, and then you don't have any surprises, and you don't have any hold calls, ultimately, if you have these pre-agreements established with the agency.
And I don't know if this is always the case, but I think in general, our experience throughout the process from pre-IND to our most recent meetings has been that the FDA at least will, spends 5 or 10 minutes at the end of the meeting to sketch out what the minutes are going to look like and make sure that the main points that everybody agrees with, and then you have those. So if you do that at the pre-IND meeting and you're all in agreement and you follow that when you come in for your IND, then they're going to have those minutes as part of the record and what the agreements were.
Yeah, that's typically for oncology. They'll do the live meeting minutes. Other disciplines may not. It's discipline specific.
So earlier we touched on lightly CMC scale up, and I think we agreed we're not going to touch on it much more than that here because it's really worthy of its own 45 minutes of panel. But Brian, what words of advice can you give on CMC scale up?
Don't discount it. It is really, it's a very hard piece to get right. They, like I said, they go on hold all the time. They're the most common reason that you'll get a rejection for your NDA application or BLA application. They have a lot of rules and a lot of very specifics. They're not flexible whatsoever in their demands. I have simple things of viral clearance. If you miss that, well, then you're going on hold for quite a long time until you get that right. So they are, to me, the hardest piece. And I think having either internally or externally experts who've gone through this process with the FDA is extremely critical at all phases of the development. And I mean, one surprising thing, while I was at the FDA, there was a drug that showed clear efficacy within for its NDA. Rare, leukemia population. The clinicians all wanted it. The CMC reviewer said, no, they're not ready. So rejection, right? That's the most common thing. And it could improve overall survival, right? But if you can't manufacture your drug, you don't have a drug. And so there was a lot of arguments, a lot of yelling about how to figure this out. And, basically, demanding that the CMC team work with the current manufacturers to get it right, which there was a high amount of reluctance from the CMC reviewers to do so. They tend not to hold hands as much as the clinical divisions will. They expect you to know your drug and how it's produced and set your critical aspects or critical quality attributes. So it's very tough.
I mean, I can speak to a recent experience where there was a small change in the supplier of one of the precursors that changed the impurity level from like 0.3% to 0.4%, and that took it out of spec, and it cost many months of going back and trying, and now doing some tox studies to rectify that issue, just to characterize the impurities that changed by just a small smidgen amount. It just is what it is, and it's something to be prepared for.
Brian, a minute ago you mentioned having the protocol synopsis at the pre-IND meeting. I just want to ask the panel, how soon do you need your protocol synopsis together?
I mean, I would put it together as soon as possible. It clearly articulates where you're going with your program, right? You think about your inclusion criteria, the population you're trying to seek, some projections of what your toxicology package might look like. I mean, you go through a lot of those mental exercises of where you want to go with your drug and where you think you'll have the most impact, right?
Yeah, and I think between regulators, investors, consultants, everyone's going to ask for that first. They want to see it because they need to understand the study to understand their piece and how it fits in. So I think as early as possible. As I was mentioning earlier, when you at the early concept of what you're going to do, it's probably a good idea to have it formulated in your mind.
Yeah, I think, as we were debating healthy volunteers versus patients for the first study, we still had early on, a sketch of what the patient study was. And the reason that we did that was exactly so that we knew what tox package we were ultimately going to do, what we were going to need, what the drug supply was going to look like in that first study. And a lot of that was important. You said, this is the indication that we're going towards the patient population. So even in the healthy volunteer study, that having that understanding was important to justify why the tox package for just a single dose escalation. This was the package that we were bringing there. Ultimately, we went to a phase 1 unit to do that study, and we worked with that CRO to develop the synopsis for the healthy volunteers specifically. So that worked out a little bit different. But we were focused much earlier on what the patient clinical development was going to look like in the first steps.
Well, and to your point about the protocol synopsis, I think it's a language that all of us understand, so it's the common language that gives us the information we need to be able to help you, to know the whole scope of your trial in a few short, paragraphs, couple of pages, honestly. So, Todd, I want to start with you. Key opinion leaders, what are your thoughts on key opinion leaders?
They can be a great asset. They can be a pain as well sometimes. Sometimes you can be a little bit further out in the science than they are. And so you may know your area better than they do, and so I think it's good always to get a gut check, but you don't necessarily have to buy everything that they say. On the other hand, as you're moving through, I think they can also be a great asset. Ultimately, you're going to need to have investigators. You're going to need patients to ultimately accept your medicine, and I think they can be a good piece of that acceptance along the way.
Patrick, where do you go to get KOLs or key opinion leaders if you don't have them?
So maybe one comment before that was, I think I agree with everything that you said, and have experienced a lot of that, both sides of it. But I think the other thing is that the key opinion leaders often, particularly at the big research institutions, they're running lots of studies. They know where the field is going, and they know what the changes are. They're meeting with other companies. And so if you come to them and say, we want to do this study, or this combination, and they're going to tell you, well, what you don't know about is that there's going to be a big phase 3 study that's going to be announced, and that combo is no longer going to be relevant in a year. So they often know from a clinical perspective of where the field's moving before clinical practice changes, and that's really important because you want to be at the front edge, not learning that you've initiated a study that by the time it completes, it's not relevant anymore. So, but then where do you get them? So for us, we came out of UNC, and so we were connected with folks there. Our other co-founder was up at Harvard, so he was connected, and then we had kind of early, say, mentors or advisors that were also clinician scientists who had moved into investor roles, and they were really helpful in the early days for helping us connect and kind of develop the protocol and think about the clinical development plan. And then as we've grown and the clinical development started moving into phase 2, then we've brought in key opinion leaders through a variety of ways. So we, through our CRO partners, the sites that they work with and the connections that they have, that's been a big help. So, we bring on a site because they recommended them and they were a good enroller and we develop a good relationship. And we've used some of those folks to come in and talk to investors and for a lot of other things. And we go back to them for advice and ask questions about the next stages of development.
Thank you. So our last topic, and we have a lot to unpack on this topic, is about operationalizing the protocol. So I really want to talk about who do you need on your team to operationalize your first in-human trial, because I think it's easy to forget all the little details, or maybe you don't know all the little details yet, like choosing an EDC, choosing data management programming, statistical programming, analytical labs, and how you're going to get the samples to the analytical labs, and how you'll provide the kits and the shipping to sites to get your samples where they need to be. Let's start with Todd, if you don't mind.
Yeah, you can start with me, and I'm going to punt it because I'm going to say that's not my area. But I know that there are great ClinOps people out there, and you need to hire one of them. And then you need to get up with some of the consulting firms who focus on those types of studies, to focus on phase 1 studies, because they know, and there's so many logistics that those of us on the scientific side who aren't very good at organization and things like planning, they do that a lot better than us. And so that's why I'm going to punt it over to those people, that just like I would if I was running a company.
Patrick, what about you? Who do you need on your study team to conduct your first study?
Well, number one, someone from ClinOps, exactly. And that was one of the, I think one of the first people we hired as we were moving towards the clinic. Yeah, the knowledge that they have and the understanding of how everything works is absolutely critical. And then from there, you can decide, all of the other functions, if you want to bring those in-house, or you're going to rely on the CRO and their capabilities, and how your specific model and what resources you have to do that really will kind of play out of that. But I do think it's critical, at the very least, to have one experienced person who's been in operations and knows all of the different pieces and how they should work.
That's such a great point. I mean, you need someone who's an expert in ICH, GCP, or good clinical practice, someone that can oversee all of the parts, an orchestra conductor, if you will, someone that can provide oversight to all of the vendors that you use and work to timelines, make everything actionable, and really be a team leader and a team cheerleader. So, Todd, I'll ask you, when do you hire versus consult versus use a CRO?
That's a good question. I think that depends on what your background is. I think I said that before, that I think I like to pull in people close to me that can cover the areas that I'm not an expert in. If it's a CRO or a consulting group that I'm really comfortable with, it's okay if it's an area that I'm not an expert in. But if it's some of the areas, like especially with ClinOps for me, not being an expert in that area, if I'm going to be outsourcing that, I want someone on my team who is a ClinOps expert that can be overseeing that, and not just trusting it fully, because it's so important to hand that over completely. So, I think you need to have a balance of what your expertise is and what it's not, and seek it out.
And I feel like you also need to understand the bandwidth. Even if you have a ClinOps person on your staff, how many compounds do you have in development? So even if they can do it, do they have the bandwidth to do it?
Yeah, and I didn't mean to say if I have a ClinOps person on my team that they're going to be doing it, but they're going to be overseeing the outsourcing to the CROs.
Absolutely.
Who are doing it.
Yeah, I think you really have options. You can do it all yourself and staff up. You can hire consultants, and I see someone, no. Or you can fully outsource it to a CRO to have everything done together as one package. Patrick, I'll ask you, when do you need to start getting proposals from CROs if you're going the route of a contract research organization?
Yeah, I think that, at a minimum, 6 months, and, you've got to have the synopsis in hand, and, I think it probably depends on the size. So, most, if we're talking about just getting started in your first IND, I assume that you're fairly small, so there's contracting and other things that needs to happen. You've got to meet with everybody. Maybe you bring multiple people in for bids. So that whole thing, that whole process takes time. And then, if you've got a target date, whether you're trying to hit that target because you've told investors or your board or whatever the reason, of why this is the date we're targeting for opening the study, enrolling the first patient, then you have to back up from there and start thinking about, well, how long is it going to take to activate the site? And what is the site? Is it, a CRO and a phase 1 unit that can get up and running pretty quick, or is it going to be a university or a handful of universities that could take who knows how long to get up and running, depending on their own internal processes, where you really have no control over that. So, I think understanding all of those things, then backing up. And I would say at the very minimum, 6 months.
Yeah, and I think if your team is very new to clinical trials and hasn't gone down this road before, and you want advice, engage a couple of companies at least a year out, because the processes we go through to even bid your study, we have a lot of boxes that we need to check. We need to understand a lot about your trial. So 6 months out from first in man is too late to start learning things that you haven't thought about.
Yeah. So that's a great point. And I think 6 months at the minimum is probably where some of our later studies are. But if you talk about that, you have one ClinOps person that's really experienced, and they're working with a team that may have clinical pharmacology or have been scientists that are now part of your core clinical team that have never done this before, and you're going to rely on the CRO, understanding how to select the EDC and build the actual questions that go in there that to capture the data. I know in some of the first studies, there were times where I'd say, well, can we pull this data? This is one of the questions that's really interesting to us. Like, oh, we didn't build the database to capture that. And I'm like, what do you mean we didn't build like that? That was clear. That was something that we wanted. But, I think as a scientist, you think about all the data that you want, but it is a whole nother art to design the system and to ask the questions in the right way to capture that data. And so if you've never done that, then it takes time. And you really, I would urge people not to overlook that step.
Yeah.
Just one thing I think that's people don't think about when they think about phase 1 units is a lot of times they're in a business and they're essentially a hotel management, and they're trying to keep the beds in the rooms full constantly. And if you come in and you're unprepared and you're not ready to go, you will miss your slot. And if you're a week late, that doesn't mean you're going to start your study a week late. It might be 3 months later, because again, if you miss your airplane, you don't immediately hop on the next one.
Those are such great points. So lastly, before we take questions, just want to get your parting thoughts. If there's nothing else that our audience hears, what's the one message that you want them to take away? Brian, let's start with you.
For a first-in-man study, you have to have your safety package together.
Know what kind of study you're going to run.
Wow, you took all the good answers.
That's why I said the middle, because I didn't know which way we were going to go.
Yeah. Safety is the critical piece, and the safety package is really predicated off of what ultimately your indication in your patient population is. So, you have to have an understanding and start thinking. You start at the future and work your way back to that point, and give yourself lots of time.
I think the one takeaway I would leave with you is that networking and coming to events like these is a great way to fill in those gaps of what you have, and meeting good people just begets more good people, beget more good people. So I encourage you to network to find the resources that you need. Okay, great, well, enjoy.